We are trying to examine the growth stage of the plants, whether they are ready for spraying plant growth regulators. So usually the most effective stage for spraying plant growth regulator is 2-node stage. So we are examining if these plants are at 2-node stage. Usually, apparently, how we can tell is if you have a 5 leaves, well, healthy, functional leaves, including your topmost leaves, usually you will have 2 nodes separated. Let's see here, and this is a detached plant here. There could be multiple nodes undifferentiated down there. We do not count. We just count the nodes with the good leaves and well separated nodes. So I removed one leaf and I can fill one node here, but I can examine by removing, peeling off the outer leaf. Apparently now, if you can see, we can see one node very clearly here. So up there, this leaf is coming from that node, and this leaf is still young. That's why we can guess that the node above it is not so much apart from the base. But there are certainly as many leaves there are, that many nodes there must be. And it is what it happens in all grasses. So here you have a node clearly. Up here, little bit separated, there is a node, and there will be cell elongation continuing in some cell division as well, because these tissues are pretty tender. So there will be cell division, cell elongation. That way this node will be separated further up. So that is how you detect like whether there are sufficient node, 2 nodes for spraying plant growth regulators. So another third way is dissecting the plants. Calvin does it very well, and so he's more skillful than me in dissecting. So when you dissect the plants, then open this, and after that you will see head formed already here. So the base of the head would be one node, and lower than that there is another node in it. Like these nodes are just trying to differentiate. That's why we may call it this 1-node stage plant. So for 2-node stage, we expect these 2 nodes which are very close to each other, they will separate little bit. So there will be more internode elongation take place. Then we'll have a 2-node stage. The internode is the space between 2 nodes. So yeah, we'll make a comparison with multiple species later.